Hi friends, welcome back to the JK Biology, and today I will be describing about the pyrosequencing. Okay. Before this lecture, I have described about the Maxam-Gilbert method of DNA sequencing and also Sanger method of DNA sequencing. So first I described that he give the pyrosequencing method. First, in the second times, in the solution based, the pyrosequencing method was given. That was the three scientists. That is, the first was Mustafa Hogi, and the second scientist was Matthias Ohlen. Third was Paul Nyren. This was the three scientists that's described about the pyrosequencing method of DNA sequencing, and this method is based on synthesis by sequencing principles. Okay, synthesis by sequencing sequencing by synthesis principle. Okay, so how this pyrosequencing process or reaction was done? You know that for the pyrosequencing or any sequencing, either maybe Maxam-Gilbert or Sanger method of DNA sequencing, first desirable things is single stranded DNA. Okay, single stranded DNA. But you know that there is a large size of the DNA is available in the human genome or any other organism, so required to the restriction digestion. Okay, required to the restriction digestion, then each fragment is used to separate in several reactions. Okay, suppose that the three fragments is obtained, so these three fragments is first denatured to find single stranded DNA, and each single stranded, that is three fragments. Okay, 1, 2, 3. So these fragments is used to another pyrosequencing process, and this fragments is used to another pyrosequencing methods, and these fragments is used to another pyrosequencing. Okay, so a small amount of this is the limitation of pyrosequencing. That's sequence only 3 to 500 base pair of sequences. Okay, this is the limitation. So required to the single stranded DNA, and how it carry. There is, when we take a, suppose that's take a DNA of single stranded. Okay, in this tube, that is the automated sequencing, but for simple understand for you, I will describe about how it will be done. First thing is add of nucleotide. Nucleotide. What is nucleotides? Either 4 nucleotides: cytidine triphosphates, cytidine triphosphate, that is alpha S. Why alpha S? Why say this is the modified form of the nucleotides of the cytidine triphosphates. D adenosine triphosphate alpha S, and D thymine triphosphate alpha S, and so on. That is the 4 nucleotides. But these 4 nucleotides is how added? Sequence wise, not including with mixing of 4 nucleotides. Sequence by first add, suppose that we add cytidine triphosphates, or maybe add single time adenosine triphosphates, and followed by another nucleotide. So this nucleotide is added, but how this is recognized? First, this is the nucleotide triphosphate is added. Okay, this was that's 4 nucleotides. Then another is ATP sulfurylase. Okay, this is another ATP sulfurylase. This was second. First nucleotide triphosphate, second ATP sulfurylase, and third DNA called first. You know that is the lino fragments. Okay, before this lecture I have described about the lino fragments, that is the DNA polymerase. First some polypeptide of the DNA polymerase is removed, of the 3 to 5 exonuclease activity, then the remaining part is DNA or lino fragments, that is called the DNA polymerase of lino fragments. This is used. If you do not know, then go back to the lectures of the Jackie biology and you can know the lino fragments. Okay, so this is DNA polymerase first. Fourth is luciferase. Okay, luciferase. Third is, fifth is luciferin. And sixth is APS. Seven is apyrase. Okay. This is adenosine 5-phospho sulfate. Okay, this is adenosine 5-phosphosulfates. What is this? Is required. Why this is required? This is the function is different. You know that. What is the function of DNA polymerase? DNA polymerase add the nucleotides. If we add, first suppose that there is A, T, C, Z. Okay. This is the simple 4 base pair of the sequences I have taken for your understanding. Okay. Then there is DNA polymerase first. Part done. Add the nucleotides. Add the nucleotides. We take only cytidine, CTP, cytidine triphosphates. Okay. That is, you know, the deoxycytidine triphosphate alpha S. Why this is used? Again, I am described about it. Okay. So, when he added, then DNA polymerase first, in that conditions, does not, this become radiolabeled, does not add, because there is not matching with C2A, because you know that A binds to T and C binds to Z. You know. Okay. A binds to T and C binds to Z. So in these conditions, these nucleotides cannot be added, and when this cannot be added, in the reactions that can harm, in the reactions, in the solutions that can harm if it is present. So that is apyrase. Apyrase degrades. Unincorporated nucleotides is degraded by apyrase. Okay. Unincorporated nucleotides is degraded by apyrase. So it's become degraded. Okay. It's become degraded. Second, we add thymine triphosphates. Thymine triphosphate, that is deoxythymine triphosphate alpha, if added. Okay. Then what conditions? It will bind to T. How it will bind? You know that there is O and OH in the 3 positions. Okay, that is lone pairs, and so there is triphosphates in the any nucleotides, triphosphates, but this OH attacks on the first phosphates of the bases, the first phosphate of the bases. Okay, here is another P, O, O, and there is another phosphates. So this there is another single phosphates. Okay, so this lone pairs will attack on this, and that will be breakdown. So breakdown, that is 2 phosphates, one and second, first and this is the third, this is second, and that was the first was before this in the nucleotides, before this in the nucleotides, that is nucleotides. There is nucleotides, there is 3 phosphates. Okay, 3 phosphates. So this attack was on that phosphates. This will release. This attack was on that phosphate. This will release. So it will release in the form of inorganic pyrophosphates. Okay, this will be released in the form of inorganic pyrophosphates if there is correct base pair added in the position A, T binds, then what conditions? Plus inorganic pyrophosphates would be released. Okay. Then the next steps will be involved for detection, for determination of the sequence which is added. Okay. Inorganic phosphate, pyrophosphates released, then plus adenosine 5-phosphate. Okay, there is adenosine 5-phosphate, APS. Inorganic pyrophosphates plus adenosine 5-phosphosulfate. In the presence of these, the ATP sulfurylase, what? ATP sulfurylase has done, it will convert into the ATP. Points out that this is inorganic pyrophosphates, APS, adenosine pyrophosphate, adenosine 5-phosphates. In the presence of inorganic pyrophosphate plus APS, adenosine triphosphate, ATP sulfurylase convert into the ATP. Okay, this is convert into the ATP. Again, when ATP is available after adding nucleotides, point out, after adding nucleotides, ATP will be found in the presence of this. Okay. Then this ATP is again ATP plus luciferin. There is luciferin that was used. DNA polymerase first add the nucleotide. Okay. And APS, ATP sulfurylase, APS, ATP sulfurylase plus inorganic phosphates involved to synthesis of ATP. Okay, involved to the synthesis of ATP. APS, ATP sulfurylase plus inorganic phosphates. Again then, there is luciferase, remaining activity of the luciferase and luciferin. Luciferin plus ATP is converted into the oxyluciferin. Points out. There is ATP plus luciferin. Luciferin is converted into the oxyluciferin in the presence of luciferase enzymes. Okay. Then there is what? Release of energy or light. Release of energy or light. And this light will be detected by the system. This light will be detected by the system. Okay. Again I am repeating, ATP plus luciferin is converted into the oxyluciferin in the presence of luciferase enzyme. Luciferase enzymes, and there is release of energy that in the form of light, and this is recognized or detected by the system, automated system. That was condition of if T is match or complementary to A. Then again another base pair is added. If there is not matching of the base pairs, that will be degraded by the A pyrase enzyme. So points out, there is 3 enzymes: A pyrase, luciferase and DNA polymerase first. That is also DNA polymerase first and ATP sulfurylase. This is the enzyme is involved. Nucleotide, that is DA4 nucleotides, and this was that. All overall, the reaction is completed, and sequence, all the 4 bases is added. Sequence. there is matching, so it is detected. Again, first we add cytidine triphosphates, that is deoxycytidine triphosphates added, that not much degraded by the apirase. Okay. Again, we add adenosine triphosphate, that is deoxyadenosine triphosphate alpha-S. What's out? This is ATP. ATP what? ATP is converted in ATP plus luciferin is converted into the oxyluciferin. So we cannot add in the form of dideoxyadenosine triphosphate. Dideoxyadenosine triphosphate. Why? This is also nucleotides. We cannot add. Why? Because if there is only deoxyadenosine triphosphate, then this convert luciferin into oxyluciferin. But in this form, we add that cannot react with the luciferin or luciferase enzymes. This form of the nucleotide DTTP alpha S or DATP alpha S. It cannot be recognized or not detected by the luciferase enzyme. So we add in the form of these nucleotides or the pyrosequencing. So nucleotide wise adding one by one. Nucleotide adding one by one, that is 4 nucleotides. If matched with the base pairs of the single stranded DNA, then the energy or light will be released that was detected by the instrument or system, and the peaks will obtained. So by adding the nucleotides, if we add, there is thymine that will match or complementary to the sequence, the light will be released. That means there is T we added, and that is complementary of A. The sequence was A here, or the sequence was T added. In the second chance, we added the complementary was A, that's the light will be released in that conditions. So it will complementary, and the second base would be a T, complementary to the A. And in that third cases, Z match and the energy is released. That process will take place, and that was detected by the systems. So there is Z complementary to C. By this method, the whole sequence can be done of the DNA fragment. Okay, the whole sequence of the DNA can be done of the single strands. But there is some problems with the pyrosequencing in comparison to the Maxwell-Gilbert method of DNA sequencing and Sanger method of DNA sequencing. Why? Because Sanger method of DNA sequencing sequences the 800 to 1000 base pairs in single time. Okay. Suppose that there is a long DNA sequences or long genomic sequences, so restriction digestion is required, and the different fragments will be obtained. Each fragment is transferred to the different reactions for the sequencing in single stranded forms, and 800 to 1000 base pair long DNA sequences will be sequenced in single times. So different reactions will take place for a longer genomic DNA sequences. But there is 300 to 500 base pair is sequenced by the pyrosequencing method. This is the drawbacks of the pyrosequencing. So repetitive DNA sequences cannot be sequenced well proper. Okay, longer DNA sequences also cannot be. It will be sequenced 300 to 500, but there is a lot of fragments required. 300 to 500 base pairs. Suppose that the human genome is many longs, that is 3 MB longs or 3 point something longs. Then different restriction and nuclease is required to digest in smaller fragments, and so there is 300 to 500. If obtained, then it will be sequenced by the pyrosequencing, but counting formation would be take place the problems. This is another part of the lecture. That's how counting is formed. Arrangements of the counting, that will be another reaction to formation of contigs. That is, you know that there is a long sequences that's required part of DNA restriction enzymes. So there is all would be the sequence by the pyrosequencing. So there is formation of the contigs. Okay. So there is a lot of DNA fragments. So lot of required the contig formations. So there is the problem to maintain or to recognize the specific sequences. So this is the problems with the pyrosequencing. Okay. So hope this lectures you understand. If you like this lecture, then please click on subscribe button and also share for your friends. Thank you.